In this video, we will discuss about the early metastatic impact of the NPY1R inhibition. So the experimental approach is quite similar that we have discussed in our previous video, where they have started injecting this Invivo 3304, which is responsible for inhibition of this NPY1R receptors on the cell surface. And the difference is that instead of continuing the experiment for 12 days, they sacrifice the mice at day 5 and then check whether it will cause some early metastatic early metastatic whether it will give some early metastatic indications or not. So what they did is at the day 1 they inject they did intrasplantic injection of 500,000 kp R172H cells, or we can say that highly metastatic pancreatic cancer cells. And after 5 days, when they check the visible liver metastasis, they are able to see significant changes between the vehicle and the actual experimental case where NPY1R is inhibited. And when they did the keratin 19 staining, they are able to see changes in the total metastatic area also, and also to the metastases that are greater than 10,000 micrometer squares. In both the cases, they are able to see significant changes in which in the vehicle the amount of total metastatic burden and number of metastases greater than 10,000 micrometer squares are higher compared to the case where NPY1R is inhibited. Then in this case, they also wanted to check what are the factors that is causing these reductions in the metastasis, whether it's the triggering of the apoptosis in the pancreatic cancer cell occurs because of the NPY1R inhibitions, or it's the decrease in the proliferations that occurs. So when they check the signature for the apoptosis, that is cleaved caspase 3, they are not able to see any significant changes in the cleaved caspase 3 signals in the vehicle and the conditions in which NPY1R is inhibited. And we can see that there are quantifications of the images of the panel E on the right hand side is shown on the left hand side of the panel E, that the signals for this cleaved caspase 3 positive cells are non-significant between the vehicle and the conditions where NPY1R is inhibited. When they check the proliferation markers, that is Ki67, what they are able to see, there is a significant difference between the Ki67 signals in the vehicle and the Ki67 signals in the conditions where NPY1R is inhibited, and the images also shows a sharp difference. So this indicates that both of these results indicates that it's the proliferations of the cells that gets affected by these NPY1R receptors, or we can see that NPY1R receptors plays a role towards proliferations of the pancreatic cancer cells, but it does not have any role towards the apoptosis of the cells. That's the reason we do not see any changes or significant changes. We do not see any significant changes of the cleaved caspase 3 signals in the vehicle and in the case where NPY1R is inhibited. The images also seems to be quite similar of these cleaved caspase 3 signals. They are not statistically significant. And in this case also, the amount of reagent that they use is 1 milligram per kg. That will cause the decrease in the NPY1R receptors. And so this is some of the last. This is the last figures that we are discussing about this paper. And overall, this paper points out that NPY1R plays an important role towards the proliferations or metastasis of the pancreatic cancer cells. And NPY1R inhibitions strategy can be used along with the other therapeutic strategies that are used for the treatment of the pancreatic cancer cells in order to suppress the proliferations and metastasis of the pancreatic cancer cells and once the cancer cells are present in a confined region, then it is easy to remove compared to the case where cancer is spread across whole body. So I hope you understood the point that they make in this paper, that is NPY1r has a role towards cancer cells, pancreatic cancer cells, metastasis, but it does not have any significant roles toward the triggering of the apoptosis on the tumor cells. That's all for this video. Thank you for your time.